Listen carefully. What I decided to do was complete chapter 10 and then pull up the review PowerPoint to point out a few things. And then I posted a pre-recorded lecture of chapter 11 to give you ahead of time because you have an exam a week from today and it's not something I want to wait till Monday to, finish completing. And then instead, what we'll do Monday is for Monday at the usual time, 7:30, we'll begin with the Chapter 11 review and any questions y'all have, and then any additional questions you have on the other chapters. Does that sound good? There's only two of you here right now at 18, so for you two, does that sound good? Okay, all right, so let's go ahead and finish up then with Chapter 10 here, okay? We had finished talking about the first of two stages that form photosynthesis, or that form photosynthesis, or photosynthesis consists of, the light reaction. Talk about how when you look at the outer edges of what is the chloroplast here, you can see the reactants you need as well as the final products you get from the overall process of photosynthesis. But when you look within the chloroplast, you see there's much more going on. So when we talked about this first stage called the light reaction, I actually separated it and gave you a kind of a formula that's nested within the general formula, where the reactants are H2O and light, and the products isn't just O2, it is a final product though, but there were two other products, ATP and NADPH. The purpose of the light reaction was actually tied into these two products here. These two products serve as energy. We already talked about ATP as the source of stored energy. And we have talked about how electron carriers carry electrons in cellular respiration, how they carry electrons to other proteins or enzymes, and that electrons, or the movement or flow of electrons, can also serve as an energy source. So you have two different types. However, we'll talk about how the electrons exactly or why they're being transported the Calvin cycle. So we left off here talking about the second stage. Second stage for the Calvin cycle. Okay, it requires a total of 3 reactants. So I had to edit the PowerPoint, and I'll show you in a second, but I did edit into your notes. It requires a reactant CO2, but CO2 is not just a reactant. As I said last class, it's actually a substrate, and I will give you the enzyme. Unlike water, which is a substrate for the light reaction, and I didn't give you a specific name of an enzyme. I actually haven't been able to find a specific name of that enzyme. Here for the Calvin cycle, the initial enzyme that catalyzes the first chemical reaction of the Calvin cycle, well, its substrate is the reactant CO2, okay? And not just one molecule. We'll talk about how this enzyme will take 3 molecules of CO2 at a time, and treat them as a substrate. But we just don't require that as a reactant, which is a substrate. Also, these are reactants, okay? But as I said before, these reactants serve as energy sources. And actually, in the case of this electron carrier carrying electrons, the electrons are going to play more of a role of altering a molecule, transforming it into, if you want to call it, a precursor that will be used to synthesize glucose. Now, the products for the Calvin cycle. Okay, the products for the Calvin cycle, well, the star product is the sugar or monosaccharide glucose. You recall the abbreviation is uppercase GLC. But we have 2 other products. The reduced form of the, I already messed it up. The oxidized form of the electron carrier in ADP+, as well as, if you want to call it, the used form or the breakdown product resulting from the utilization of ATP, ADP plus P. The I stands for inorganic because they often call it an inorganic phosphate. Never heard a biochemist, add any importance to it other than it's just nomenclature. So often we weren't even required to write the subscript I. We just put P with a circle to indicate a functional group called the phosphate group. So you have a total of 3 products produced for the Calvin cycle. But only one of those 3 products, glucose, is a final product like oxygen is, okay? So you only see these 2 in the formula. So you got to break it up as you go through this. Now for the Calvin cycle itself, okay, it occurs not in the thylakoid membranes, but the Calvin cycle occurs in that viscous solution, which is depicted in gray here, called the stroma. It is also light independent. Well, that's why they didn't put light in the name of this, but they put light in the name of this, but it's light independent. Chlorophyll molecules, and therefore light, energy do not play a role in the Calvin cycle. Purpose of the Calvin cycle, to produce a sugar, glucose. Okay, and we're going to talk about how during the Calvin cycle, a process called carbon fixation is performed, whereby the atoms of carbon in the molecules of CO2 are covalently bonded together, resulting in the formation of a carbon backbone. Well, that carbon backbone is going to end up being the backbone of an organic compound called glucose. Okay, CO2 is considered an inorganic compound. In actuality, there's 5 types. We didn't cover them in detail, but there's 5 types. Okay, but this, as I said last class, this is a key area of interest to scientists. Okay, now, in order for the Calvin cycle to occur, however, all right, there are enzymes as well as products that need to be produced that require the electrons as well as the source of energy called ATP to help power this Calvin cycle. And well, it's coming from the light reaction as it shows you here. Okay, so the purpose of the light reaction is actually these two here, to produce these two. So here's that formula for photosynthesis. Again, we talked about last class. What did I say this molecule, what did this represent here, this molecular formula? What did I say last class it represents? This here. Anybody? Anybody? Right, glucose. Now, all monosaccharides have this molecular formula. They're structural isomers of each other, but this actually represents glucose. Okay, but the reason for putting it this separate way for the Calvin cycle in particular has to do with that process called carbon fixation. Okay, so if we rewrite this formula, okay, applying it only to the Calvin cycle, all right, I still have, let me get this to work with me. I still have my CO2. Of course, it's going to be 6 here. Okay, and then over here, I still have my glucose coming from this formula up here. However, if I'm rewriting the formula specific to the Calvin cycle, on the side where we would put the reactants, I have to add in ATP and an ADPH from the light reaction. Over here, I have to add in 2 products, the breakdown or used product of ATP, ADP plus BI, and then instead of the reduced form, the oxidized form of the electron carrier, denoting that when it exits the Calvin cycle, it's been oxidized. It's no longer carrying electrons. Now, the Calvin cycle is broken up into 3 phases, okay? The first phase, phase 1, is called carbon fixation. Carbon fixation is carried out by an enzyme called Rubisco. Okay? In other words, Rubisco catalyzes a chemical reaction called carbon fixation. The second phase is reduction. Well, here's that word reduction. So it must have something to do with what, everybody? If the second phase is called reduction, what does it probably have to do with? Why would they call this second phase reduction? I know it's early, but I got to ask you questions so I don't hear, y'all don't doze off on me. Why would we call it the second phase reduction? What does that probably have to do with? Yeah, gaining electrons. So in other words, this is where our electron carrier comes into play. But if you notice during phase 2, this is also where ATP comes into play. Okay, so all this for phase 2. So as you go through this, if you notice they're trying to change the color pattern here for phase 1 leading into phase 2, and then 3 changes not much, but there is a little bit of a difference in color between these two. But reduction is primarily due to this is where the electron carrier comes into play. Okay, so we'll go over this where the electron carrier, the reduction is not applied to the electron carrier. Okay, the reduction is applied to the molecule that's going to regain those electrons. Okay, because we'll talk about how at the end of phase 2 we get this molecule. Next, second only to Rubisco, okay, and Rubisco catalyzing a chemical reaction called carbon fixation. Second only to that is this molecule here. Okay, so this is, I guess you could say, the co-star of the show. The third phase, well, you can title it two ways. Okay, the third phase is titled the regeneration of the CO2 acceptor. Okay, well, everybody, the CO2 acceptor is better known among biochemists as RU. Okay, we're almost there. This should be a B, and this should be a P. Okay, I came close. Didn't mean to try to make the R look fancy or anything. Okay, but RUBP. Okay, so where it says regeneration of the CO2 acceptor, you could rewrite it as regeneration of RUBP. Okay, that's the third phase. So these are the three phases we're going to go through. There are molecules you're going to have to be able to recognize, but the test is going to be multiple choice, so just make sure you can recognize them. I've given them to you in your notes in a certain order also. Okay, so phase one of the Calvin cycle. Phase one of the Calvin cycle requires a reactant, or more specifically, a substrate called CO2. Okay, but it not only requires CO2, the substrate, it requires an additional reactant, our CO2 acceptor, RUBP. Okay, so we have a total of two reactants, but only one serves as a substrate that are required by an enzyme called Rubisco. So the abbreviation for enzyme, which I believe I gave you in chapter 8, is just ENZ. Okay. Now in your notes, I gave you the long name for Rubisco. Rubisco is actually abbreviation, or if you want to call it, how do you say, in general, an acronym. It's a long name. It's there in your notes. I just gave it to you because, like I said before, I have students that want to know, what's the longer name. But everybody, 99.9% of the time, it's just called Rubisco. Okay. So Rubisco needs these two reactants, but it treats CO2 as a substrate to perform a process called carbon fixation. Carbon fixation is a process whereby the atoms of carbon and carbon dioxide are covalently bonded together, okay, creating a carbon backbone. Now, the molecule that's actually produced that contains that carbon backbone is called 3-phosphoglycerate. So this is the product produced by Rubisco. Okay, I'm not going to write it out, but I'm just going to put an R for reactant over here, and over here I'm going to put down, this is an R for a reactant, but I'm going to hyphen it for an S because it's more specifically the substrate. Okay. All right, so again, the R stands for reactant, and then if I put a hyphen S, that means more specifically it's a substrate. Okay, but if you notice, it says here, what Rubisco does is it takes 3 molecules of CO2 at a time and takes those total of 3 atoms of carbon and covalently bonds them together. But you can't see a molecule here necessarily in the beginning with just 3 atoms of carbon covalently bonded. What you see is this large thing, and each charcoal bead is representing something separate. Each charcoal bead is representing an atom of carbon, and the lines are covalent bonding. What it is, everybody, is RUBP is called a CO2 acceptor. The best way I can think of describing it is if you're especially somebody that likes to build things at home, or you're into kind of arts and crafts, carpentry or arts and craft, things of that nature, you normally need a what to work on in order to create what you want, a workbench. Okay, so in order for Rubisco to perform this process called carbon fixation, it uses RUBP as its workbench. So they call RUBP the CO2 acceptor because these molecules of CO2 that are undergoing carbon fixation at one point are chemically or covalently bonded to RUBP. But what Rubisco is actually doing is using RUBP as a workbench to create that carbon backbone. Does that make a little bit of sense, everybody? Be honest, unless you dozed off. No, yes? Okay. Man, I need to start off with something loud that screams at y'all. Okay, so what ends up happening is Rubisco uses RUBP like a, but it's called a CO2 acceptor because initially these atoms, carbon from carbon dioxide, are being covalently bonded. But the end result is we do get a molecule with a 3-carbon backbone. That's a product called 3-phosphoglycerate. Okay. Now, just to let you know, all right, because you do need to know this is the final product for phase 1 carbon fixation produced by Rubisco. I'm not going to test on this specifics, but just to let you know, okay, the 3-phosphoglycerate. The glycerate, okay, is in reference to a molecule with the 3-carbon backbone. The phospho is in relation to this. This is a phosphate group, one of 7 functional groups. The 3 has to do with chemists number the atoms of carbon that form a carbon backbone. So this is carbon 1, this is carbon 2, this is carbon 3. The phosphate group is covalently bonded to it, thus 3-phospho, but the overall 3-carbon molecule is a molecule of glycerol, but they call glycerate. Just to let you know, now I'm not going to test you on that high, that specific, but just to let you know where the name's coming from. Then what happens is this 3-phosphoglycerate will enter phase 2 called reduction. Again, there's only a couple of enzymes total that I really want you to know their specific names. But again, overall, the Calvin cycle consists of a series of chemical reactions catalyzed by a group of enzymes. So there are more enzymes in phase 2, but there's only one I want you to know, Rubisco. But as this product that exits phase 1 called 3-phosphoglycerate enters phase 2, there are enzymes, okay, that act on it as a reactant now. And these enzymes require the energy of ATP. But when they utilize the ATP, breaking it down to ADP plus P, they take that phosphate group that they broke off and they attach it to the other end of what was once 3-phosphoglycerate. Now the molecule still has what is considered a backbone comparable to glycerol, and it still has phosphate groups, but now there's not one, there are 2, thus the bis, B-I-S. The 1 and the 3 just have to do with the first carbon and the third carbon. I'm just letting you know where the name comes from. What I want you to know is that 3-phosphoglycerate, which is the product produced at the end of phase 1 by Rubisco, is suddenly the reactant to begin phase 2, and it is converted to 1,3-bisphosphoglycerate, utilizing ATP not only as a source of energy, but also utilizing the phosphate group of ATP to help transform or convert this reactant to this product 1,3-bisphosphoglycerate. Then what will happen is there are other enzymes that come into play, and they will take the electrons from NADPH. In other words, they will oxidize NADPH to NADP+. And when they oxidize NADPH, okay, NADPH is losing its electrons. Thus, we rewrite it in its oxidized form, NADP+. But what these enzymes are doing is they're transferring the electrons to 1,3-bisphosphoglycerate. This molecule then, when it gains 2 electrons, is reduced to the final product of phase 2, glyceraldehyde 3-phosphate. Okay, so how is this final product produced? They're in the last chemical reaction where 1,3-bisphosphoglycerate was reduced, okay, using the electrons from NADPH, thus converting this reactant to this final product. This molecule, glyceraldehyde 3-phosphate, is simply and most popularly is known as G3P. Going into the test, know it as G3P as the final product of phase 2, reduction. Now what happens here is they're actually giving you little numbers. Now I don't get real specific with the numbers a lot of times, but they're actually giving you numbers as far as how many molecules of this are produced, how many molecules of this are produced. That's what these numbers are referring to. Basically what happens is by the end of phase 2, okay, beginning with 3 molecules of CO2, by the end of phase 2, we don't have 1 G3P produced. We have a total of 6, okay, for every 3 molecules of CO2 that go into this Calvin cycle. Of these 6 molecules of G3P, on average, 1 will actually exit the Calvin cycle and be converted to glucose. Now it says glucose and other organic compounds, and I have a lot of students that for whatever reason don't get what it means. Okay? G3P is directly converted to glucose. It's the glucose that's used to create other organic compounds. Okay, that's the way we normally put it. What I want you to know going into the test, though, is that G3P, a small amount, or 1 out of every 6, will exit the Calvin cycle and be converted to glucose. From there, the plant can use it as chemical energy, or it can use it to make other organic compounds to simplify it. Now, the remaining G3P, or simply the majority of the G3P, stays in the Calvin cycle and enters phase 3, okay? Regeneration of RUBP. Okay? So now what happens is that the majority, or 5 out of every 6 G3P molecules produced for every 3 CO2, the majority enter phase 3, and with the help of enzymes, the G3P is treated as a reactant, converted to the product RUBP. RUBP, which is our CO2 acceptor. Okay? But notice, in order to do that, it requires not only the energy of ATP, but again, it requires the phosphate groups of ATP. So by the end of phase 3, this CO2 acceptor known as RUBP has been regenerated. Okay? What's the importance of regenerating the RUBP? Because Rubisco cannot begin another round or catalyze the first chemical reaction to begin another round of the Calvin cycle unless it has its workbench, unless it has RUBP to help stabilize when it creates that carbon backbone using CO2. So in summary for photosynthesis, okay, we have our light reaction. The purpose of the light reaction is to produce chemical energy. Okay, type of energy-rich molecule ATP, as well as electrons that are going to be needed for the Calvin cycle. However, we talked about sometimes what happens with the light reaction, okay, is that first of all, the light reaction involves electron flow. And we talk about sometimes what happens with the light reaction is these electrons that are flowing flow in a cyclical manner. The purpose is extra ATP. However, whenever the light reaction occurs, usually these electrons flow in a linear manner, or they take a linear path, and that's the one what we want, that the plant wants the majority of the time, which happens to occur the majority of the time, so that these two products are produced. Now there's O2 that's produced, but it's not needed by the Calvin cycle. And then the Calvin cycle, well, it uses that chemical energy in the form of ATP. It needs the electrons from NADPH. Okay. Notice here in parentheses, I put ATP plays a role in phase 2 and phase 3. NADPH, the electrons it's carrying, plays a role in phase 2. And then the Calvin cycle produces G3P, which is then converted to glucose. What's the purpose of photosynthesis? To produce the nutrient glucose, which happens to also serve as chemical energy. Okay. Now I know that was quick for the Calvin cycle, but what I want you to know for the Calvin cycle, I pretty much redid the notes last spring and then again in the summer to have basically what I want you to know going into the test. Because like I said, it's a lot of biochem. Now, are there any questions on this before I pull up the PowerPoint to review? I know it's early in the morning. Let's see. All right. Let's see here. So for this review, I left off more details compared to Chapter 11. Chapter 11, I'll pull up on Monday, the review. Okay, so for anybody who showed up late, just to let you know, what I'm going to do is I'm just going to cover Chapter 10, cover the Calvin cycle today and the review for Chapter 10. I'm not going to go into 11. What I'm going to do is post a previously recorded lecture on Chapter 11, so you have all the information ahead of time going into the weekend, because you have a test, an exam a week from today, and there's a lot of information to study. But on Monday, I will review Chapter 11 using that review that I posted. And hopefully that'll help out some. So make sure that you definitely, before Monday morning, before we have lecture on Monday morning, that you go over that, okay? So, but for chapter 10, this is the review. So if you notice, if when you study your notes and you look at the PowerPoints, this is basically the figure that I'm feeding off of, okay, for the majority of the lecture. So from the very first slide, I didn't put it down, but what you want to do is whenever you see these figures, try to label these figures. Well, with this figure, where you want to start off with is label everything outside of, hold on. What you want to do is label everything outside of this double line. These double lines here are supposed to represent the outer and the inner membrane. Therefore, the overall structure, even though there's no arrow pointing to it, what label do you still want to add? Even though there's no line pointing to this, what label do you want to add to remind yourself what this overall structure is? What would you write in down here? Should be pretty simple, not a rhetorical question. So this whole figure, right, you just want to write it in. So it's not there, you just want to write in chloroplast, okay? The gray area here, what would you want to write in, even though there's no, how do you say, box or anything to specifically say label this? What would you still probably want to write to let you know what this gray area represents? Okay, y'all answered the other question, so I know y'all are awake, some of y'all. Not matrix, that's mitochondria. But for the chloroplast, what would the gray area represent? It's okay, Gomez. You gave me something, okay? That's why I'm asking. It's described as a viscous solution. And I said, don't get it confused with pores called stomata, because this one has an R in it. What's that viscous solution located? No. Chlorophyll's not a solution, it's a molecule. I'm glad I'm asking this question, although I'm getting a little depressed. Okay. All right. Okay. How about if I give you the ST? Okay. We'll do this like y'all ever played Hangman. Okay. Not stomata. Stomata, I said, is the pores in the plant leaf. I said, don't confuse it with stomata. I said, this one has an R in it. Okay, thank you, Stroma. Okay, if anybody's being driven crazy here, I have to ask these questions. So I'm squeezing it in so these letters are running into each other. So on some of these figures, and this goes for all your chapters, there might be areas where it actually doesn't say or point out, you label this, but you need to be intuitive enough to know, well, I still want to label it, okay? All right, so Angelina, speaking of thylakoid, where in this figure would we have the thylakoid? Would it be these circles? Would it be this gray area? Would it be these little green disc looking things? I'm giving you only three choices. Yeah, the green discs. So again, they don't mention it here, okay? And the stroma, stroma I put out here, these right here, you'll probably want to label out here. And you would put the thylakoid, I'm just going to put a T. You would label the thylakoid out here, okay? Now, why is it I don't want to write anything directly here or write anything directly here? Because I want to leave room for what? The two stages, excuse me. The two stages, okay? So where would I write the first stage? Would I write it in the gray area or would I write it in the thylakoids? Would I write it in the stroma of the thylakoids? If you're labeling this, where would I write the first stage? Yeah, in the thylakoid. Okay, so I would write the first stage here. What's the name of that first stage? There's already a hint on the slide, everybody. It involves light, so you should know the first right. Light reactions. Okay, remember this is where light comes in. Also, what's unspoken? Why do they color the thylakoids green? Why is it depicted in green? The stroma is just gray. That's just to make it stand out. But the thylakoids in this figure, they're actually coloring them green for a reason. So that's another thing you need to notice. Why are they colored green? What do you find in the membrane of thylakoids? Yeah, but what do we find in the membrane that has to do with light? right? Chlorophyll, right? It doesn't, yeah, there you go. So combination of the two, Rendon and Gomez. Okay, so they're depicted as green because in the membrane of the thylakoids are chlorophyll molecules. Thus, this is where light energy comes into play. But we have two reactants coming into play here. Up here would be the second reactant. What would I write in here? Right, H2O. So these two here together collectively are our reactants, but only one of the two serves as a substrate, which would be H2O here. Okay. Now we talked about, now you'll have some questions about what's going on, okay, but if we're going across the top here, it's showing you two of your three reactants. So as far as the edges go, the outside of the chloroplast, you take that formula I give you for photosynthesis and you write the reactants, two of which are substrates outside. Yeah, yeah, it would be. But I'm sure there are minds out there looking at that. So if you look here, down here at the bottom, this is where you would take the products from that formula for photosynthesis and fill them in. So if you're labeling this, you probably want to start outside, then work your way in. Then from there, you would have your light reaction here. But over here, you would write your second stage. What would you write in here instead of a number 2? What would you write in the center of the circles, or the circular figure they give you? First stage is a light reaction. What's the second stage? All right. And then you just fill in everything else. Okay, you would just fill in everything else, okay? Now, I give you some questions here, all right? So if you notice the first couple of questions, you can actually go back here and associate it with that. So labeling that figure should help you out with these first two questions. What is the formula for photosynthesis? Well, that's everything you list here and here. That all has to do with the formula for photosynthesis. List the two stages of photosynthesis. Well, I just said you would list the two stages here. I just put 1 and 2, but you would write the actual stages. Then it starts talking about what are the reactants required for each stage, what are the products. Again, you go back here. If I'm just looking at the light reaction, one of the products would be listed down here because it's the final product. The other two products would be listed over here. Well, what is this first product that's a final product that exits the chloroplast? What would it be? It's derived from H2O. When 2 electrons are stripped from H2O, that breaks down H2O to what waste product? Something tells me y'all prefer I just lecture and do the talking. No, 2, there you go. Over here, though, notice these two products stay in the chloroplast, indicating they're not final products, they're going over to the second stage, the Calvin cycle. So this right here, what would you write in here that I'm circling with the finger? What would you write right here? All right, and what would you write down here? Yeah, NADPH. Okay, so these questions, I put them all on here, but they're actually related to the figures. Okay, what is a photosystem and what is the function of a photosystem? Well, this here. First thing you want to be able to do is label this figure. I'm telling you there's a photosystem here. Well, but you actually have to take a step back. What I'm not asking you in the question is, again, what you have to be intuitive enough to know that you need to do. What does this actually represent before we ever get to the photosystem? If you don't know, where would you look in order to label this figure? What I put the bracket around, what does that represent? Okay, let's take a step back. Yeah, bilayer, but of what? That's right, phospholipid bilayer, but of what? Is this the phospholipid bilayer of the cell membrane or plasma membrane? Or is it actually, right, the thylakoid membrane? So listen carefully. The fact I ask you a question on the photosystem, first thing that actually needs to come to mind, okay, is the stage that you're in. When we talk about photosystems, which of the two stages are we referring to? There's only how do you say, of the two stages, there's only one of those two stages that we talk about photosystems. Which of the two stages? Right. Okay. And again, going back here, the light reaction occurs here in your thylakoids. Okay, that's where we will find photosystems. So let me ask you again. So what does this represent then? If we only talk about photosystems when we talk about the light reaction, and the light reaction occurs in the thylakoid, what does that phospholipid bilayer actually represent? This is how I write reaction Rx in. We said photosystems, the only time we talk about them is with the light reaction. We said the light reaction occurs in the thylakoids. So what must this represent then? Has to do something with the thylakoid, right? What part of it? Phospholipid bilayers form what, everybody? So they form the plasma membrane, they form the membrane of membranous organelles, right? They form membranes. So this is a membrane, but of what structure? No, just a membrane. I said, what do they form? Not function, but form. Yeah, this is a thylakoid membrane, everybody. Okay. The photosystem itself is this thing here. Okay. What you'll want to do then is you'll want to label the purple parts. You'll want to label the blue parts. Okay. This blue center right here, what do we call it? What is it called? It's the center of the photosystem. I'm actually giving you half the name. Okay, I'll let y'all figure that out there. Make sure you go over that. Then what the next question is going to ask you, okay, is what is the function of a photosystem? Well, you can see here they're showing you arrows moving, little beads moving. Well, something's happening. This is occurring. Okay, so make sure you know whenever we talk about linear electron flow, again, which of the two stages are we talking about, everybody? Maybe I'm not conducting this correctly. I don't know, or maybe y'all haven't studied enough. Y'all are going to wait till the last second. Yeah, my summer class found out that despite that first multiple choice test, the second one wasn't as nice, and it was multiple choice. I told them they were thankful that we didn't meet in person because it would have been a lot more brutal. Make sure y'all study, everybody, get ahead. So basically, what is the function of a photosystem? This is going to be tied into electron flow and the production of ATP and NADPH. It also asks here, do the electron transport chain and ATP synthase play a role in photosynthesis? Your answer should be yes. That is highlighted here. And that's what this is representing. But if you notice up here, okay, this here is to represent what I'm asking you about the ETC and ATP synthase here. Okay, what is this trying to tell you about the ETC and ATP synthase? That they not only play a role in cellular respiration in the mitochondria, but they also play a role in what else? Not a rhetorical question. In other words, do we talk about the ETC and ATP synthase in both cellular respiration and photosynthesis? Yes or no? Right? So what I'm trying to point out to you, okay, what I'm trying to point out to you is that when you learn the function of the ETC and ATP synthase, during oxidative phosphorylation and cellular respiration? Well, the role is comparable, okay, to what occurs during what? Photosynthesis. You shouldn't treat it as something new. You should already know it by the time you see it again in chapter 10. You should have already studied it in chapter 9. So it asks about how. That's what that figure's for. So that's basically this one here. It has to do with question 4. Okay? And then the last one is just the, I'm sorry. Then I gave you this here. This is the one. What is this representing here, everybody? This right here, along with this internal space, all of this is representing a piece of a what? Right. Okay. So then this gray area here, what does this gray area here represent that's surrounding the thylakoid? Not the space. It was colored gray in the first figure too, and I told you what it was. Remember I told you it's not stomata, right? It's stroma. There you go, stroma. What are these things right here? What is this one here? And what does this one here represent? What do they represent? I gave you a figure where it just showed them in the membrane. So what do they represent? Not reaction centers. I'm circling the whole thing. The reaction center makes up part of it. Right, photosystems. Okay. What about this in here? What does this represent? That's connecting the two photosystems. That's bridging the gap. Right. And what does this represent down here? In particular, this line here connects to this line. So if this is the ETC and they show a red line coming here and going here, what are they probably trying to show you? That the ETC is tied into what? ETC is a network of proteins or a protein complex. And what it does helps an enzyme do its job. So what I've circled is an enzyme. What is the enzyme? Its name comes in part from the product it makes. It synthesizes a certain product. Right, ATP synthase. That's what this is going to represent an ATP. So this is that figure I showed you where it gives you a wider view or wider detail. Okay, up here. So if I circle all of this here, okay, I've just circled everything associated with the light reaction. Okay. If I circle everything here, I've just circled everything associated with the what? The Calvin cycle. Is this helping at all? It's tough when you're not keeping up with your studying, but is it helping at all? Okay. That was my main concern. And then this is just the Calvin cycle. Okay, pretty much you're just going to be filling in the boxes and knowing the steps, and then you're going to feed off of your notes in the PowerPoint to help ensure that you get everything. Okay, and then these boxes here are where you will write in the phases. Okay, so that's the boxes where you will write in the phases. Okay, and then this is where you will write in the enzyme and its substrate. And then down here, as well as here, you'll write down what is the product produced by the end of phase 2, and then down here what it's converted to. So that's just to give you an idea. Okay. Are there any questions